very many of you before. I've come over from England for padding for this. I work at a place called the St. Lab, which is in the east of England, in Norwich. It's about an hour north and east a bit of Cambridge. It's not too far from the Sang Centre. And today I'm going to talk about the work we've been doing with some GMOD tools to try and stimulate the opening up of sharing of data and info in a small genomics community and the impetus behind all that. Okay, so I'll kick off with this graph. This shows global hunger index. This is the number of people in a country that are living without enough food and therefore most at risk from things like famine due to changes in the food supply. There are constant pressures on our food supply, I'm sure you're probably aware. Things like ever-growing populations, climate change, shifting land use, and sudden changes in food supply are caused by lots of things, some by wars, some by natural disasters. And all these really lead to severe and sometimes deadly shortages that mostly affect poor people in developing countries, but they have effects in our part of the world, over here in the West. I don't just mean the West of the USA, but the West of the world in general. Global food shortages will manifest as price hikes, and we end up with situations where governments have to dole out things like food stamps in their own countries. One of the major threats to the stability of our food supply, one of the most unpredictable, is the emergence of new plant pathogens. These are basically diseases that suddenly appear on our radar that we had no idea about before. An example of one of these is a pathogen called UG99, a strain of stem rust. This is a fungus that devastates wheat. It's spread by spores in the air. And it first emerged in Uganda in 1999, and it's spreading out and heading east towards this region in India. This part of India is the third biggest wheat producing region in the world, and it's a very poor region, and a lot of the population is in danger from food poverty. We don't have any way of dealing with UG99, so the potential for the loss of crop supply there and the disruption of food security is massive. And as I said, plant disease, the emergence is really unpredictable. We don't know what might hit us, and whatever's next could be devastating, and we really need to be able to move quickly as scientists to be able to deal with these diseases. And I think it's important to try and realise, actually, that science has been traditionally very bad at responding to a quick change in situations. The usual model that we have is quite inhibitory to a joined up and fast response, actually. This is because the normal way of working we have, we rely on high profile publications to get further funding. So we sit on our results until the last minute until we can make the most of them. And we release the data only when we publish. So to save our own skins, we end up chasing the money, basically. And this is perfectly fine, right? Because we all need to have careers, we all need to feed our kids. It's not the wrong model, because ultimately scientists spend, we don't directly make money. So not every project should have a right to exist. I'm not arguing against the funding model that we have. I think competition is important and necessary. But really what I'm trying to say is in situations where speed and collaboration really are paramount, then the model can let us down. So if we can come up with a way of responding quickly, we stand a much better chance of responding properly in emergency situations. So we need to stop messing around with the standard way of doing things when it comes to emerging plant diseases. We need models of working that can get us going as quickly as possible, outside of the usual way of doing things. Essentially, this is going to mean getting basic analysis of genomic resources like sequence and related data sets created with little or no central pool of money. And related to this is the idea that genomics overall, as well as being big business, is nowadays, for a lot of groups, much more of a cottage industry. Thanks to the massive drops in costs and the success of tools for assembling and annotating genomes, much smaller groups are taking on projects than is traditional in this field. So there are lots of communities with draft level assemblies, the annotation of which would benefit from mechanisms for bringing together the knowledge being generated in different parts of those small communities. So one quite big opportunity to try and rework our way of doing things came in the UK at the end of 2012. A disease of ash trees called Ash Dieback appeared in Norfolk. This is a disease caused by the filamentous fungus Hamniscyphus pseudoalbidus, goes by the other name Cholera, through the vagaries of naming You see on the slide is a typical symptom of Ash Dieback. You can see this symptom on the leaves throughout the summer. You see weeping lesions on the stems and branches throughout the year. And in the spring you can see fungal fruiting bodies popping out of tissues throughout the season. And these symptoms are often lethal in the trees, so it's potentially devastating. The disease was first reported in Poland in 1993, so it's been coming across for about 20 years. There's still not been much impetus to do things about it. But it was first noticed in the UK in February last year. No more than 300 cases confirmed across the UK, and up to 90% of 80 million ash trees are thought to be at risk. So we have this fast-spreading outbreak of a potent pathogen. Scientists, would like to think we should be able to do something about it. And over the last couple of decades, political situation and damage to tree stocks, like the loss of elm trees, Dutch elm disease, and other agricultural disasters, like foot and mouth episodes in cows, has sensitised the British public and the government to tree losses. And when Ash Dieback hit, there was a public outcry and decided to act quite quickly. So we were able to throw away the typical business model of working and trying some things that would maximise our speed. We thought that this would help with the current problem of Ash Dieback, but hopefully feed back into some of the issues of food security and to novel species analysis as well. So Ash Dieback is our test case for new ways of working. So to try and make the most of people and data power, we're trying a crowdsourcing approach. So crowdsourcing has quite a lot of definitions actually. It's different in everybody's hand, but basically it's a practice where we make, in our case, whatever data we can generate and find instantly available before we analyse it ourselves, and we let whoever can help do whatever they can with it. And in our approach we crowdsource, not just from a particular set of scientists, we try and do mathematicians and bench scientists. We hope that the practical upshot of this means that the valuable data is going to get to people much more quickly, and we can basically speed up the research cycle. Another advantage of this, putting a science out there, is that the results are examined and filtered in a live peer review process that speeds up the whole thing without having to wait for publication in traditional model journals. So, try to apply the best of open access data principles to try and fast forward collaborations on genomics, and we put up a hub website to facilitate this. It's called Open Ash Dieback. It's at this URL. I'm not going to show you any screenshots of it. The important thing is that the data and the results themselves are actually hosted on GitHub. Probably everyone in this room is familiar with GitHub, right? Anybody not know what GitHub is? Okay, I won't bother to explain it. But we use it because it's really great at tracking changes to files and keeping track of who made the changes and exactly what the changes were. And we think this is going to be critical for the proper attribution of contribution. So we chose Git as the sort of central hub of our crowdsourcing effort. Basically, GitHub sits in the middle and tracks the changes of whatever we're doing and provides the framework for merging the conflict. It's the centre. So we made all the Ash Dieback genomic data we generated available via GitHub, completely free and without any sort of forward license. And we've received quite a lot back in return. So we've been able to push quite far. We were provided with a DNA sequence and first draft assembly from TGAM, which is a sequencing centre in Norwich, and that's what we can see here. We've got quite a good assembly, first draft assembly. We've learned quite a few things, so we can see from the assembly, for example, that the genome has a somewhat complex structure, atypical of parasites, an expanded genome structure. These peaks in the pink track correspond to collapse repeats. So we see an awful lot of collapse repeats, which is typical of pathogens. We also got analyses of the genome for genes that are typically involved in lignin and cellulose degradation. These are the major components in wood tissue. The genome was compared with those from a load of fungi that are capable of directly rotting and metabolizing wood, and some typical degradation genes were made. So over in this end we've got the numbers of lignin and cellulose degradation enzyme. Up here the basidiomycete rotters, and Cholera, Ash Dieback appears down here. So it looks like it doesn't have a complement of genes, for example, for getting rid of or decaying wood, so it's not likely to be carrying out that function. And as we had donation also of strains from around the UK that we went on to sequence, we were able to do SNP calls relative to the reference that was created and show the provenance of the different strains across Britain and across strains from other countries. We've got the emerging hypothesis now that the Japanese isolates might be very distantly ancestral, and that the organism might be drifting in from the Far East. Okay, so that's what we achieved by putting raw data out there on GitHub for bioinformaticians. And that's fine, but bioinformaticians have a habit of doing bioinformatics analysis and knowing a bit less about the hardcore biology of the pathogen than they might. And we wanted to try and capture that sort of information. We realised that to try and really add value, then the important thing was bringing the bench biologists who know all about the pathogen. And to do that, we had to sort of liberate the genomics data and the expertise to them and get the insights back from them. And I think that crowdsourcing in this sense, wide-ranging and hopefully sustainable crowdsourcing, is going to be dependent on the community that we can build. So the whole task that we have sort of generalises to one of how you build better biology and bioinformatics communities, and one big part of that is how you incentivise it. So, as bioinformaticians responsible for this data, the first thing we tried to do was saying that any other bioinformatician work for SALT would have done, was to put together the basic databases and tools with the right query interface that would help biologists find what they were looking for from their bit of sequence, or their gene or protein or function or the name gene, and let them to start interrogating the data that was being created and uploaded. So naturally, we used GMOD tools for this. This is the analysis part of the stack that we put together. It's based on GMOD stuff and common open source tools. So the main thing is Web Apollo. It's the major genome browser style component. Golem is a Git-based wiki engine, very lightweight, and this enables us to have every object in the database, in the Git database, to be annotatable through the wiki. And we use Sequence Server, which is a Ruby gem that implements a BLAST server for sequence-based searches. And all these are tied together, and I kept talking with GitHub through a piece of middleware that descended from an internal text-based annotation tool we had called GFoo. This thing still carries the name GFoo. It acts as the dispatch engine for data to Web Apollo, and acts as a feature linking engine for the wiki and sequence store for the BLAST server. It coordinates automatic exchange and concurrency of data between the different components and the different bits of database. So making sure that contributions from all these other tools are up to date with each other and tracked into the GitHub repo. It also provides a RESTful interface to the repo data. It's basically a Rails app to take advantage of the Rails built-in routing system to provide a RESTful interface. And as one other interesting feature in that, as it's adapted from an old lab tool that was for multiple strains, then we can work on dispatching data to multiple strains within our Git, even the multiple organisms, so we can enable to coordinate data from the Ash and from that didn't render at all, did it? Looks a bit suspicious.
A little bit.
Yeah. Don't squint too hard. But yeah, from both organisms, the pathogen and the host. It runs on a couple of other bits of software that develops. It runs on BioSAN tools, Ruby gem, so that we can directly incorporate BAM files. It runs on, for creating small static pictures, it actually runs on a Ruby gem called BioSVGs, which generates genome browser style pictures. It doesn't generate them interactively, the static SVG style pictures. And it also incorporates Thomas Downs' biodalliance as a portable viewport within dynamically generated web pages. So the stack allows us to provide data and gives us a way to take in data while we're at it. GFruit just provides this central way of maintaining it. Okay. But when it actually comes to getting the contributions from the individual specialists, then we have quite a different problem. So first of all, I think the biggest hurdle is the publish or perish imperative that we have in science. So as I mentioned, scientists really need publications. At the moment, pretty much the only recognised research output is the paper, which is why people sit on the results. But as the world gets more and more progressive, open science movements are emerging and pressing on and having some sort of effect. So we're exploring the opportunities that might be coming about now to start valuing much smaller pieces of research in different ways. And I think that this will be, again, one of the other keys to effective crowdsourcing. We think that people will do it because they want to help, but we think they'll keep doing it if they feel that they're getting the right sort of credit, and we need to start looking at mechanisms to foster the behaviour. So we've been implementing reward badges like these into our tool stack. So I bet you're familiar with badges. They're sort of tiny virtual rewards for completing particular tasks. I bet you've got them on your Facebook page for bits of games, or in your PlayStation 3 games, or even on little fitness apps on your smartphone. I had one send me a badge that looked like a birthday cake for going for a run on my birthday. Really nice. But Mozilla provides a very flexible and very useful open badges scheme. So as a contributor, you can, through Mozilla's open badges scheme, you can earn badges for accomplishment. That's sort of the basic thing. But it also provides tools for issuers. So it provides a framework for you to certify the content of the accomplishment, the skill, so that then the contributor can display a verified badge. It's not just a bit of a graphic, it's actually verified in some way, and I'll explain that in just a second. This is how Mozilla Open Badges work in practice. So as an issuer, you need to basically bake your own badge. So a badge is a PNG file with some JSON metadata stored within it, and this metadata contains information including something called an assertion. That's the description of what the badge is for in the form of a URL or a web page that describes that in plain text. It also carries the name and a unique ID for the person to who the badge belongs. You can't just take your badge, give it to someone else, print it out. You can't steal badges under this framework very easily. So as an owner, what you can then do is just display the badge on your own website, in your CDP, via something called the Mozilla Open Backpack, which contains and provides and manages the URLs for all your open badges. Mozilla provide the Backpack API and metadata spec, so it's very easy to create and manage these badges. So we added two very specific tools to our stack to manage badge awarding as contributors use the tools. So the first of these is called Merit. It's a standalone web app for actually managing the badges. We also added a second one called Scoreboard, and this is a tool for tracking the activity of a user within the apps and issuing one of the badges through Merit once the achievement has been reached. And the whole reward stack is actually built on quite a number of technologies. This was all basically coded by Martin, who sat at the back, and he likes to play with languages. He's polylingual, and to try and make everything interact as easily as possible. The whole stack is fully RESTful, so its communication is very easy. Okay, so Merit, the app for managing and issuing the badges, is very easy to install and use. It's a simple form-based interface for creating a new badge. You just need the PNG file with the basic badge design and the URL with the assertion, and you're good to go. All you have to do through this web form is type in the email address of the person getting the badge, their name, you select which badge you want to give them, hit the button, and they're awarded the badge. Nice and easy. Scoreboard is a JavaScript Web Apollo plugin that sits and listens to a user's activity within our tools, and it maintains a small side database of specific sorts of activities. These are the definitions of the things that you get the badges for. So any particular activity that relates to a badge, it listens for. So for example, within Scoreboard, the badge issuer specifies the rules by which badge should be created, and once some sort of threshold is passed, it automatically messages merit, which does the issuing of the badge. So you might have a badge, say, for editing exons. When somebody edits 5 exons, or let's say adds a protein, all the steps along that path are recorded within scoreboard, and the badges are issued as soon as they're earned. And the user is kept informed of progress by little pop-ups within the tools. So things like this pop up within Apollo as you're editing the tools, and little click-throughs just allow the badge to be collected. And they'll go straight into the user's Mozilla backpack. Straight. Okay. So we actually provide quite a few badges. For example, give you an example of some of them. Contribution gets you straight away the Open Scientist badge. Editing features gets you the feature editor badge. And there are lots of different levels of each of these badges. So you can go stepping through every U5, every 10, every 20, you get different iterations of the badge. And submitting large-scale experiments like an RNA-seq data set can also get you a badge. So I should provide these for our training as well. You can see a full list of the badges that we issue at the moment on that web address. So, how many badges have we issued with the set of tools so far? We haven't done any yet. This is mostly an artifact of timing. The crowdsource repo's out there, and this web tool's just coming to fruition. We're actually waiting on, there's a little bit of politics going on. We have to wait for some Ash data before we can actually publish it. I'm sure you're familiar with the problem there. But we're pretty sure it will start to pick up steam in the next couple of weeks, and we'll start to see the community contributing. So the community, that the first instance of the software service is actually still very young. We're a little bit ahead of the curve, so we're not discouraged at this point. As well, I'm not a big believer in build it and they will come. So I wouldn't have started this without some sort of market to sell it to, and I think they will come. Just parenthetically, our training program at the same lab uses badges, and in the first, I think, 3-4 months of that, since that's been running, we've issued 70 badges along their side. So people do seem to want to collect them for skills attained. Let's hope they want to collect them for work done as well. So it's quite an exciting time. So what's more and gives us more hope is that other projects are already keen on making use of the arrangement of tools that we've developed. So a much more mature community, the wheat yellow rust genomic community, are keen on getting their hands on this because they want to do annotations and just try and slurp all that data into a database. Similarly, I would like to finish a talk on a disgusting slide. A very new project looking at the genomics of the recent invasion of Britain by the Spanish slug. Actually, you can't see that slide. It's horrible on here. I promise you. It's 3 slugs fighting.
I'm sure it's terrifying.
Yeah, it's horrible. Oh, it makes me take looking at. Yeah, this project about the Spanish slug hopes to use this as well. Okay. So, very last thing, because we're going to try and use it for other projects, this software stack, we needed to make it as easy to install as possible. So, you can get source code for all these projects from GitHub if you want source code, but if you just want to try and install it, we've made the whole stack deployable through Docker. I don't know if you know Docker, it was new to me, Martin told me all about it, but it's an open source project to create lightweight and portable containers from any sort of application. And this little coupler should enable you to get an instance up and running wherever you want it. And if, I used to say this to biologists, but if you do want to give it a go and you don't have the staff to set it up, I'm sure everybody here does, we also will come out and help you if you like. Okay, so just a couple of quick acknowledgements then. So most of the work that I presented here was the coding work done by Martin, who's signed back and has a poster just on the left if you want to go and see it. A couple of the guys in my group, and we get one of the badges that we have were designed by a recent graduate graphic design student from the Norwich School of Art, young lady called Abby Bolton. And we're funded by the Gatsby Charitable Foundation and the BBSRC. That's all. If you want this talk or any of the links in this talk, they're all at this URL. Thank you very much.
Cool.
Questions for Dan?
Coffee time, right?
It kind of is. Yeah, because my first question was I wanted to hear all about the data. I wanted to hear about
Right.
how many people were getting badges and stuff.
Yeah.
I know.
Just that too much early. Next week, maybe.
Right.
Yeah.
Well, I'll definitely be following along. I think it looks really fun. I think so. Badges are great. I'm wondering if we'll need something more in terms of real life, like T-shirts. Bitcoins would be nice. I was thinking T-shirts for like the top 10 badge earners or something.
Yeah. I mean, I think this will appeal mostly to younger scientists.
I have no doubt that
undergraduates,
that's who it's going to appeal to. Yeah.
Postgraduates, but they're the ones at the bench, right? Most
Yeah.
of the time.
Yeah,
They're
yeah.
the ones writing the lab book, so hopefully they'll think to write it down in 2 places rather than just 1.
Yeah.
Tenure for the top 10 badges.
Can you talk a little bit more about how people actually display the badges? Are you essentially completely delegating that to the backpack concept?
Yeah, that's what it's for. Yeah, the Mozilla Backpack acts as a central location, so people can collect badges from me, from anybody else, right, from any organisation, and they have them in one central place. But they get a URL for that, or they can download an image, so they can deal with their badge however they want to. So they can reference it, they can embed it in their web page, they can import it as a picture into their Word CV if they need to.
I was just wondering if there's a way to actually bring that display of those badges more within the Web Apollo framework. Somehow, as you edit, you can see who's editing around you, and you click on them, right? Because that type of...
Yeah, that's...
Well, I was going to say that too, actually, yeah. Yeah, so that you can see who's winning right now. What's the top 10, or 10 might be too many for...
So we need a trophy cabinet, right?
Yeah, exactly,
Yeah.
yes.
That's somehow integrated.
Yeah, that's it.
Yeah, I mean, screen real estate is at a premium in Web Apollo, but something small would be good.
Maybe even just a little trophy icon up near the file.
Yeah.
Is it fair to hire slaves to earn badges for you?
Yeah.
Well, I thought that's what we were talking about. Were we talking about graduate students?
With things like that, you have to let your own moral compass be the guide.
All right. Well, very good. So, according to the schedule on the wiki.